Well, good afternoon, Fikri. How are you?
Good, Richard. How are you?
Doing well, thank you. And just as I was saying before we went on air, I'm so excited and happy that you and I are doing this today. And it's spring. It's really a great time of year to be working on these kind of things. So thank you so much for joining us. Thank you again.
Thank you.
Yes. So welcome to Rare Awareness Radio. We are here with Fikri Birey, and this is part of an ongoing series that we have had for a little while now, where we have been exploring CACNA1A, and sort of had lots of different folks come on and talk to us from the physician standpoint, the caregiver standpoint, the scientific standpoint. So I'm really excited to have you come and speak with us today a little bit about CACNA1A and the work that you've been doing. I know you have lots of interest in many other areas as well. So maybe we'll just start there. Maybe we'll just start with a little bit about your background, a little bit about who you are, and sort of what kind of started driving you towards disorders and genetic therapeutics.
All right, of course. And first of all, thanks for having me on. It's a pleasure to be talking to you today.
Yeah.
Um, Originally, I am from Cyprus, and I came to the U.S. in 2004 for my higher education, and
Mm-hmm.
that's really when my scientific journey started. Um, Throughout my training that span, I guess, a couple of decades now, um, I've been involved, I guess, in developmental neurobiology, neurogenetics, stem cell biology, all in the context of disease.
Mm-hmm.
So that was my main driver all throughout my training. That I was very interested in understanding how disease emerges, particularly in development, and how it culminates into uh, what all of us recognize as a disorder of a certain kind.
Mm-hmm.
So, towards the end of my PhD training, where I was studying stress related disorders and how different neural subtypes interact in mouse models where we were trying to recapitulate some aspects of stress. um, I got really captivated by this emerging technology at the time, and what it could mean for us to understand disease and what it could mean for us to treat disease. And this actually is a Nobel Prize winning technology now. It's been termed as reprogramming.
Mm-hmm.
And just very briefly, what this technology enables us to do as researchers is to take a, skin biopsy or a blood from any individual and quote unquote reprogram these cells to assume a stem cell like state. And what that really means for us is that we can then take these stem cells and coax them through various means to any cell type of interest. For us, we study the brain. So for us, those are neurons or other non-neuronal cells in the brain. So what you can imagine this gives us is an individual cells with the exact same genetic makeup, but and then their neurons without having to really go into inside their brain and getting those neurons, which is something that you cannot really do. Um, so this really excited me towards the end of my training. I really wanted to
Mm-hmm.
continue my training in this area. So I joined Sergio Pashka's lab at Stanford for my postdoctoral training, and that's where I really met calcium channel disorders, and that's where my really fascination about this particular set of proteins and the diseases associated with them really started. Um, and in Sergio's lab, I studied Timothy Syndrome,
Mm-hmm.
which is a calcium channel disorder caused by mutations in a very similar calcium channel gene to CACNA1A, CACNA1C. So I use this stem cell, we call them this reprogrammed stem cells, induced pluripotent stem cells. And so I developed this models, the now called organoid models, these three-dimensional iPSC or induced pluripotent stem cell derived models that resemble certain parts of the brain. And we develop models of the brain regions that we think are responsible or where the disease manifests itself in Timothy syndrome. And through my work there, I really came to kind of develop this appreciation of the impact of not just Timothy syndrome of the that has on the patients, but calcium channel disorders or calcium channelopathies
Hmm.
really have on these patients at large. So when I started my lab here at Emory in 2022, I really wanted to continue on this line of work. And what really, I'm just taking you step by step here. What really took us, our lab to CACNA1A is really our interactions with the patient advocacy group, CACNA1A Foundation,
Right.
that really galvanized and excited us to really focus on this one particular calcium channelopathy as part of my initial research directions.
Wow, it's quite a run up from your beginnings in Cyprus all the way to opening your own lab at Emory. I
Mhm.
want to drill down for a second, because a lot of folks listening might not necessarily be clinical. I think when people hear calcium channels, they might think heart, they might think calcium channel blockers. I'm
Mhm.
really glad that you mentioned both the work that you've done with Timothy syndrome, and by the way, congratulations on your your Nature cover story. And I'm really glad that you also mentioned the 3D human cellular models that you've been working with and the role that they have been playing and how you've been sort of looking through developmental therapeutics and what advantages they offer. Can you just, for the layman's perspective, Fikri, can you talk a little bit more about the calcium channels and what role they play here?
Yeah, absolutely. So calcium channels are we can think of them as literal channels
Mhm.
that shuttle calcium in the cell. And calcium is an extremely important um, secondary messenger or a signaling molecule. You can also think about it that
Hmm.
really, depending on the amount and through which channel it comes to the cell and various other means, really signal a whole series of things to the cell
Hmm.
about its survival, about its activity. It's extremely important for neurons to fire. It's extremely important for certain genes to be activated. So it's um, calcium is life itself in a way. So it's that's why even minuscule, tiny changes in the amount that it gets into the cell or how that process is regulated, any kind of perturbations there can really kind of snowball into
Hmm.
bigger and bigger dysfunctions for the cell. That's why these channelopathies can really lead to quite severe consequences for the patients. Um, we really focus on the neurobiological consequences, but as you mentioned,
Hmm.
the heart is also very much affected in a lot of these calcium channel disorders.
Hmm.
Timothy syndrome, it has a very significant component, cardiac component, in how it clinically manifests a lot of arrhythmias,
Yeah.
tachycardias, and things like of that nature.
Hmm.
So it's a multi-system disorder, and basically, in terms of the human cellular models that we developed, we really wanted to understand the impact of the mutations on this calcium channel gene, um, not just in neurons that have been grown in a two-dimensional setting, which has been the kind of traditional cell culture
Mm-hmm.
means of going from stem cells to a particular neuron of interest. But we really wanted to take that next step where we grow these things in a three-dimensional environment that resembled the region, the brain region that they belong to. So for us, that was the cerebral cortex.
Mm-hmm.
So I worked, quite a bit of effort in the initial stages of my training went into developing the models without even having to think about the disease.
Mm.
Our first question was, how can we develop a three-dimensional model where we can really model some of this phenomena that relates to the development of the cortex that you cannot really model with any other two-dimensional system. And that included migration of certain neuronal types, and after the migration, their integration into this network that already existed, and then them forming this interconnected networks that resemble the first cortical networks. To be able to follow all of these processes, it had to be a 3D model, and then we developed this not only a single region but two-region human cellular model that we ended up calling assembloids. And that was really critical for us to really understand some of the core phenotypes that we then discovered with Timothy syndrome.
Wow. There's been so much, Fikri, that's promising in sort of the advancements of genetic therapies. And of course, you mentioned the work that you're doing with 3D human cellular models and the role they play, and understanding rare diseases. Today, there was an announcement about some personalized gene therapy, right? That seemed to have some significant advancements in its ability to cure a state within, I think it was CLP, right? If I remember correctly in the article I read. Are there other sort of promising advancements in genetic therapy? Do you think that all comes to CRISPR and sequencing and personalized gene therapy at some point? I'm curious as to what you see are the most viable sort of treatments and in conditions, especially for things like Timothy syndrome and CACNA1A.
Yes. I mean, that's a great question. And it's, there are probably a number of valid and slightly differing answers to this question as well. My own perspective is that a lot of the rare disorders, and I think a lot of the people will agree in this space, that has been quite difficult to treat and therapeutically target because of a number of different reasons. One, a core advantage over some of the more polygenic or diseases like schizophrenia, for example, where we cannot really triangulate onto a genetic factor. We don't really know the gene. Is probably a lot of small effect mutations on a lot of genes that kind of culminate into that sort of a disorder. On this, on this side of the table, the rare disorders where we do know the genes responsible, but yet, there are other complicating factors for the mutational spectrum, right? Like CACNA1A has thousands of different mutations associated with it. And number one challenge there is to really understand the impact of these mutations. It's not a given, so it's not just because we know which single base pair mutations are there doesn't really tell us about what it does to the channel. And really understanding that is a huge undertaking if you really want to understand all 4,000, 5,000. And even if you understand that, then making one size fits all therapeutics for something with such heterogeneity is it's extremely difficult.
Yeah.
And then beyond that, we always talk about delivery as well.
Right.
So, like, when do we deliver? When is the right time? How can we really access the right cell types and right region? But having said all that, that even though these are the challenges, we at least know what the problems are, which is not really, for a polygenic multi-site disorder. Sometimes you don't even know where to start.
Right.
We know the problems at hand, and I think you mentioned that kind of a very critical CRISPR based therapy that came out today.
Mm-hmm.
There are some other ones that are kind of flying a little bit under the radar that we see that are coming up. Kind of NL1, it personalized therapeutics that have been developed for just that one patient, that I find extremely exciting. That shows us at least that even though these are somewhat costly and time-consuming and labor-intensive processes, that means that it is within our reach. That we can develop therapeutics, whether those are ASOs or CRISPR studies, that really can that are curative, right? So that they can go and fix the thing at its origin. That's so seeing that this is possible. I think that it's just the problem comes down to scale and safety, and that's where I think organoids also come into play a little bit more, and we can talk about that also.
Sure. To me, it's so many things, right? It's inspiring. It's amazing. It is also a little daunting, right? We've been talking about personalized medicine for years and years, and well before the CRISPR gene and other therapeutics that were out there. And to see some of this coming to fruition now, like you said, even with all of the caveats and the challenges associated with it, it is rather remarkable, isn't it?
Exactly.
Yeah. A little bit ago, you had mentioned Sergei Pashkov, and when you first came and became a part of what they were doing at their lab, and honestly, I've talked to so many folks like yourself where collaboration seems to be such a key part of the work, right? Because there's so many aspects to this. There's so many very specific expertise that are often required. Talk to me a little bit about, your sort of approach to partnerships with researchers, collaborations. How do you make all of that work for you, Fikri, as you try to navigate what can be a very beneficial thing, but also can sometimes be a very complex thing?
Yes. Given how modern science operates, I think how interdisciplinary things have become, how it's absolutely crucial, for anything that will be cutting edge or impactful, to have many hands on it. So I think that's as I was a postdoctoral fellow in Sergio's lab, and Sergio was the first to actually derive human neurons from induced pluripotent stem cells for Timothy syndrome. So he had done the initial phenotyping in his lab, and as a postdoc, I think he had started and continued in his lab, and then I kind of continued that work in 3-dimensional models when he started his own lab. My work, my work spans kind of this initial phenotyping, and then we were at a place that we thought we kind of understood enough about the biology of Timothy syndrome that we could start nominating various genetic therapeutic approaches, and that's when the ASO work started taking shape. Yeah, and then that's when I started collaborating with another postdoc in the lab, Shayu Chen, who really just was a fruitful collaboration in kind of bringing the know-how about the phenotypes and kind of genetic therapeutics, molecular biology know-how, and kind of really ended up being a extremely synergistic and fruitful collaboration at the end.
Yeah, that's outstanding. I love to hear that. You mentioned the CACNA1A Foundation, right? And the Buffalo Initiative. I don't know if you're also doing anything affiliated with them, but certainly these advocacy groups, these patient communities, they play such an important role, right, in shaping research, treatment. Tell me a little bit about how you interact, I guess, Fikri, with the CACNA1A Foundation, and maybe also, I think folks might be interested to know what your sort of interaction is also with patients and families.
Yeah. As I said, foundation was critical in us really prioritizing CACNA1A as one of the main lines of research in my lab. And CACNA1A Foundation is an extremely proactive organization that they have their hands on research, advocacy, communication. They're kind of helping fund early stage research. They're building natural history studies. They're connecting patients with researchers, holding the symposium conferences where everyone gets together and really brainstorms and prioritizes things based on needs of the patients. And it's from a researcher standpoint, it's extremely informative in how it guides our research. It's extremely fulfilling. But it's there's also, I guess I would say, the sense of urgency that we feel that we don't necessarily feel with some of the other studies. So in that way, it's been extremely critical. And I think they played a big role in now how we're thinking about the next steps with the project, where we're also developing ASOs for a particular patient, and our further collaborations where we're also kind of trying to move this to preclinical models with in vivo models, and it has given us this heavily translational lens through which we view this work.
Interesting. I had mentioned the Buffalo Initiative, and just for your benefit, Fikri, that's a separate organization that Sunita Malapati and some folks from the CACNA1A Foundation have started to try to accelerate drug therapeutics. So they're trying to look at it a little bit more from the sort of the venture capital aspect of it, because I mean, look, at the end of the day, funding is everything, right? I mean, to so many of these things for all of you. And I mentioned a little bit about patients, Fikri. I'm kind of curious, as families are affected by CACNA1A related disorders, they're trying to navigate the research landscape. They're online. They're, they're consuming all of this information. What advice do you give those families to kind of make sure that they're looking in the right places and getting the right information?
Yes. I mean, I would say, first of all, that they are not alone. So that's the first thing. But there's a lot of advocacy groups, patient groups that work around these rare disorders. And, I mean, everything starts with a genetic screen and then a diagnosis from a clinical geneticist. And you might get a name of a gene that the geneticist thinks is responsible for the condition. And more likely than not, when you put that gene and foundation or advocacy and you Google that, you'll come up with an advocacy group that is associated with that gene. I would say probably the first step is to engage those groups. And when it comes to engaging researchers like me, I mean, I'm always thrilled and motivated and excited when I hear from patients through emails or anything else, and or whether that means explaining our science or connecting them with the foundation myself, or anything around that. More than happy to do, and I'm sure I'm not the only one in this. Um, and I would say the last thing probably is to stay hopeful, because the science is really moving faster than it ever
Yeah.
has. And the involvement of patients, where you've been a patient or part of an advocacy group, or you're a new patient, um, and they're just sharing your story, I think.
Yeah. Yeah, that's a really good point. Just sharing your story really matters. And I appreciate too what you said about like don't be afraid to engage and reach out, right? So important. And the science is moving faster than it's ever moved before,
Hmm.
right? And I know for a lot of families it still probably doesn't feel like it's moving fast enough, but
Yeah.
it definitely has increased in speed. And that kind of leads me, I'd like to talk to you a little bit too, speaking of as we are moving very quickly now,
Hmm.
what kind of, from a futuristic standpoint as it pertains to genetic therapeutics, you've talked a lot about the work you have been doing, which feels very futuristic, but kind of what's next for your research? What are you most excited about? And as you kind of look ahead, Fikri?
Yeah, it is really exciting and a hopeful time
Mm-hmm.
for rare disease research, rare disease therapeutics research. And I really do believe that we are at a pivotal moment
Mm-hmm.
where these technologies that have been maturing for a while now are starting to converge,
Hmm.
as they're reaching a point where they will be finally empowering this novel therapeutic development. And whether that's patient-derived models that we're using, that's precise genome engineering, or oligonucleotide therapies that we're now seeing more and more examples of,
Hmm.
um, or whether that is really developing models to really understand the impact of these variants at scale, so that we don't have to rely on doing this mutation by mutation, but we can do 1,000 mutations at a time. And that's
Wow.
an area we're also very, very much interested in developing in the future. Uh, but so altogether, I think these are coming together to really put us on a kind of, on a momentum
Yeah. Yeah.
to really get to a place in this space that we haven't really been before.
Hmm.
As for us, I think we are, as I mentioned, we're very excited um, to really see through the CACNA1A work, and really see the initial series of studies that we're doing. Number one, what are some of the new things that we can understand about the channel itself, about
Mm-hmm.
about channel when it's harboring these mutations from patients, to really get some novel insights. um, and then just to bring in this work that, empowered by our experience with the Timothy syndrome, to really kind of meld that with this phenotyping that we're doing,
Mm-hmm.
and really to see what the next step might be there. Like
Right.
we've done all the in vitro work. Now we are collaborating to see what the in vivo, in an in vivo model, what our shortlisted ASOs might look like. But then what's the next step after that?
Mm-hmm.
And that will likely involve, again, a cross-disciplinary effort that will involve maybe the industry, that maybe involve other advocacy groups that you mentioned, venture capital, who knows.
Who knows? Yeah. But
but
many people. Yeah.
but yeah, so I'm very excited to see how much we can push that, uh kind of like the bench to bedside,
Yes.
kind of a plan forward.
Yeah, and the basic science is so important, right, Vickery? I mean, the bench science still remains to be so important here. And then I think about what you're saying about the in vivo to eventually getting to, a therapeutic, and then human trial, and it's just there's so much, right? And to
Mm-hmm.
your point, there's so many different interdisciplinary aspects to that because everybody brings different pieces of expertise as you move down further and further down the path of an actual therapeutic that can be consumed by a human, right?
Exactly.
Yeah, that's wonderful. Fikri, before we let you go, I just want to thank you. Make sure that there isn't any important point that you want to make sure we get across here today. But the work that you're doing with Timothy syndrome and CACNA1A related disorders and the impact that you're having on patients and families one day at a time is just so impressive. So thank you for that.
Thank you, Richard. It was a pleasure to chat.
Yeah, I really enjoyed it as well. So, folks, we've been speaking with Dr. Fikri Birey. And once again, thank you for your time. And we'll look forward to speaking with you again soon.